we have Pano Therapeutics.
I'll kick it off just by expressing some thanks, gratitude to the Connexum Catalyst MetaBCD team and community for having us on. I think we first met this community in October 2019, and it's been incredibly supportive of Pano since we started on our entrepreneurial journey here. So the Pano tagline is that we're targeting mitochondrial complex I to unlock the potential of pan-therapeutic drugs. So we'll explain both this target cohort and this concept as we get into the materials. By way of personal introduction and the presenting and broader Pano teams, I'm Ben Gibson, former recovering investment banker. I've been working with Francesca on building Pano for about 3 years, really with a great focus on mitochondrial and metabolic health, with the perspective that this is really one of the biggest global problems right now, and a very important area of focus for human health across a bunch of different areas. Francesca Fieni is the co-founder and chief science officer. She's a mitochondrial biophysicist who made the core discovery of a novel target around which Pano is being built at UCSF about 4 or 5 years ago. And the most recent addition to our internal executive team is Ken Batchelor, who's our head of R&D, who's previously the CEO of Novotarg, which was folded into Pano about 6 months ago. And prior to Novotarg, he was the SVP R&D at GSK, who was responsible for drug discovery through human clinical proof of concept for drugs in metabolic cancer and antiviral areas. It's my pleasure to announce today that Maury Birnbaum has just joined Pano's board of directors and our scientific advisory board as chair. Maury was most recently the chief science officer of Pfizer's internal medicine unit, and before that was a research clinician or research physician and professor at University of Pennsylvania, where he really focused on areas of very key significance to Pano, metabolic health, and Biguanides. Joan Manick and Thomas So, who are very active on this community and this conference, are also on our board. Joan is the CEO of Tornado Therapeutics, and Thomas, of course, is the CEO of Connexum Services. We've put together a really remarkable scientific advisory board of global KOLs, as I mentioned, now chaired by Maury. If we get to the end of the presentation with some time, we'll go through their bios in more detail. So Pano's executive summary is really our vision is pan-therapeutic mitochondrial drug discovery and development. What we mean by pan-therapeutic is a common set of therapies with a common target positioned or optimized to treat a broad range of diseases across different therapeutic areas. Very apropos for the theme of this event. As I mentioned before, it's really built around the discovery of a novel drug target by Francesca. We call it mtTOB, sort of a play on words on mTOR. It's the mitochondrial target of Biguanides, physiological regulator of mitochondrial complex I, which Francesca will explain. And we really believe from a drug discovery perspective, the knowledge of mTOB and our ability to manipulate mTOB differentiates the therapeutic, broadly therapeutic play topic versus the toxic mitochondrial complex I inhibitors, which is an age-old mystery. On the basis of this discovery, we've built a platform for drug discovery and development that enables the first rational drug discovery of mild and transient complex I inhibitors, otherwise called partial complex I inhibitors, which is really the effect that we seek. And Ken Batchelor brings to the table a platform for OCT optimization of Biguanides for tissue targeting and penetration, which is also one of the great elements that's required for discovery and development in this area. In terms of drug development, we're initially focused on a cluster of rare and high prevalence kidney indications. We've got some very interesting in vivo data, and IND enabling studies are ongoing. In terms of our development strategy, we're going to be focused on internal development of a cohort of rare orphan kidney indications and moving towards partnering for very high prevalence kidney indications associated with metabolic syndrome. Francesca?
Yeah, so basically I'm going to give an overview of mitochondrial complex I, which is essential to energy metabolism. Indeed, it contributes 40% of the mitochondrial automotive force, and thus it's a rate-limiting step for oxidative phosphorylation. It's also the main site of reactive oxygen species in our cells and possesses two well-characterized conformational states, the active A state and the deactive D state of complex 1. So the D state of complex 1 occurs during abnormal metabolism when substrate availability is compromised and during ischemia. So about a 5 to 50% fraction of complex 1 is physiologically in the D state in our cells. So next slide, please. So there's more than 60 family of compounds that inhibit complex 1, and although attempts to try to classify them have failed, we can still cluster them in two main buckets. There's the mild and transient complex 1 inhibitors, whose class prototype is metformin, and then there's a toxic and irreversible complex 1 class of inhibitors whose class prototype is rotenone. Of note, rotenone and metformin do not have the same mechanism of action. Ben, can you go back? Sorry, I think you pressed too fast. So this mild and transient inhibition is very relevant because it modulates oxidative phosphorylation and ROS by activating indirectly AMPK and suppressing mTOR. So this class of mild and transient inhibitor has a pan-therapeutic potential across a range of diseases. However, the mechanism of action and the molecular target of this mild and transient complex 1 inhibition has been elusive or at least highly debated. Of note, metformin preferentially binds to the D state, the deactive state of complex 1. So next slide. So this is the target discovery based on electrophysiological characterization of electrical currents across the electron transfer chain in the inner mitochondrial membrane. So we discover a novel potassium uniport across complex 1, which, as Ben mentioned, named mtTOB, mitochondrial target of Biguanides, as it is the target of metformin and clinically relevant Biguanides. So mtTOB supports the transition to the D state of complex 1. And so Pano's thesis is that mtTOB is physiologically transient, but chronically activated in the abnormal state, metabolic state. So as I mentioned in the previous slide, metformin preferentially binds to the D state of complex 1. And this is very important because it's already been confirmed by 3 independent groups. And I cited 2 papers here that just came out in August 2022 and September 2022. And this is basically, one of this is a nature cryo structure of complex 1 bound to Biguanides. So thus we can fairly say the mild and transient complex 1 inhibition is associated to the D state inhibition and to mtTOB inhibition. So at Pano, we're going to capitalize on the knowledge around mtTOB modulation to basically enable, hopefully, the first rational drug discovery of this class of compounds. Next slide, please. So these are, again, electrophysiological representative recordings of mtTOB and its pharmacological modulation by clinically relevant Biguanides. As you can see here, there's metformin, fenformin, and proguanil. However, canonical Biguanides are not optimized for activity in mtTOB, nor they are at tissue-specific activity, which is mediated by OCT, the organic cation transporter. So again, we're going to capitalize on combining the knowledge around mtTOB biology and selective OCT activity and tissue penetration of a novel class of Biguanides that inhibit with this mode of action. And Ken is gonna basically give us more details on our R&D programs and the diseases that we're gonna pick in Pano. Thank you. Ken, up to you.
Thanks. So it is the combination of inhibition of mTOB with our novel Biguanides, which can be optimized for activity in target tissues. And you'll see on the slide, we can make best-in-class complex 1 inhibitors with a very wide range of therapeutic indications. Different tissues have different expression of these organic cation transporters, and we can design compounds to go into different disease areas. We are focusing initially in kidney diseases, and this is an area where metformin can't play. It has a black box warning for use in patients with impaired renal function, and our compounds are designed to be safe and efficacious in kidney. Next slide. So here you see the combination of effects. Looking now at Pan 1195, we show that it is a potent inhibitor of mTOB. The K channel that Francesca described, which can be optimized for use in the kidney, where OCT2 allows uptake in the kidney and OCTN1 and OCTN2 support reuptake in the kidney. This gives excellent exposure of drug in the kidney and good efficacy, but very importantly, reuptake prevents elimination of drug in urine and increases safety and reduces the risk of lactic acidosis in patients with impaired renal function. Next slide. So our compounds have ideal PK profile to treat kidney diseases. They are stable to metabolism, have long half-life, good bioavailability and exposure. Importantly, good exposure in kidney, obviously the target tissue, which you will see at the bottom where metformin has none. The other really important thing is that our compounds are not eliminated in urine where metformin is, which of course creates the risk of lactic acidosis. And we've confirmed these data, you'll see on the right at the bottom. In dog, you see the same thing there, that metformin is eliminated in urine. Our compounds are eliminated in feces. Next slide. So we've done a lot of work in polycystic kidney disease in mouse models of various types. And here I'm showing an experiment with 1238 in polycystic kidney mice, dosed for 7 days with 50 mg per kg. And here what we show is proof of mechanism. We show activation of AMPK in PKD mouse kidneys. We show inhibition of phosphorylation of S6, consequences of biguanide pharmacology. But we also show, importantly, that kidney weight to body weight is reduced by 27% in just 7 days. And in this experiment, there is no effect on body weight or other organ weights, and 1238 was well tolerated. So 1238 is an interesting compound, and it's one that we intend to progress further in the treatment of polycystic kidney disease. I'll pass it over to Ben.
Thanks, Ken. So just picking up there, as Ken mentioned, we've got two great lead candidates focused on this cluster of kidney indications, broadly broken into rare orphan kidney diseases and high prevalence kidney diseases. We're going to be running both lead compounds side by side through a battery of in vivo protocols for both of these subgroups of indications, with a view to selecting one or both to go forward after IND-enabling studies that we expect to complete in 2023. There's very significant unmet medical need in both the rare and high prevalence subgroups of this cohort of kidney indications. And really, if we just look at the main orphan kidney indications, there's well over a $5 billion peak sales opportunity and very significant unmet medical need. We've also got some very interesting in vivo proofs of concept for female reproductive cancers and an opportunity to plant a flag with an oncology application or oncology program here in due course once we've advanced the kidney cluster. In terms of our foundational IP estate, we've licensed patent covering the assay for the modulation of the novel target from UCSF. We've got issued USPTO composition of matter IP around a novel specification of biguanides that covers our leads, and we've got other families of composition of matter IP coming up behind them. As we mentioned, we've put together an incredible group of global KOLs on our scientific advisory board and our board of directors, really covering the metabolic health, mitochondrial biology and genetics, as well as the various therapeutic areas and specific indications in which we've got interest. Maury Birnbaum, we discussed, Doug Wallace at CHOP in Philadelphia, Michael Kaplan, the chair of the Cellular and Molecular Physiology Department at Yale Medical School, Ralph DeFranzo and Zan Fleming, who are responsible for the clinical and regulatory development of metformin, the canonical biguanide, for its main application in type 2 diabetes, and Michael Pollack and Fred Hauscher, who are really metabolic medical oncology experts who are going to be supporting the launch of our oncology program in due course. So with that, I'll turn it over for questions.
Thank you, Ben, Francesca, and welcome, Ken. You and I actually spoke a few years ago. You're down in at the beach in Carolina, are you not?
Yes, and I'm going to apologize now because I don't know, I don't remember that.
It's okay. It was a few years ago and it was once, not to worry.
I look forward to reconnecting, so that's good.
Super, super. Well, that brings us to the end of our time. So what I would like to suggest is best of luck. It sounds like you are taking a unique approach to go where others are not. And kidney is near and dear to my heart, given my own therapeutic exploration history. But Ben, Francesca, Ken, best of luck, and we'll be watching.
Yeah,
Thank you
you.
very much. Appreciate
Thank
your
you so
support.
much. Thank you.